This is a production of Cornell University.
Cool. Thank you so much for having me. Got two stories I'm going to tell today about these Tn7-like elements. So I don't think I need any explanation for who this is. Still the only woman to win an unshared Nobel Prize in Physiology and Medicine to this day as a woman. So still something for SIPs to be very, very proud of. So obviously she's the patron saint of transposons, discovering them, winning the Nobel Prize in 1983. So you may recognize this harvest corn as one of the areas where the movement of these elements could turn on and off the color genes. So this was the ACDS element. Wait, I gotta use this. Cool. So TN7 and all the TN7 family elements are also these DNA transposons. They move always through DNA, never an RNA intermediate, which is another large class of transposons. And how they move a transposase, in this case it's TNSB. This is the equivalent of the transposase in Barbara McClintock's ACDS element. It recognizes these two ends of the transposon, so that's how it knows who it is. Everything between those two elements is what moves. So it synapses that together and it goes to a new location. So what's exciting about TN7 that differentiates it from other elements is its level of control. So most transposons are basically moving randomly without much selection for target sites. TN7 is the exact opposite of this. This is what got me amazed about it some 30 years ago, and I've never not been captivated by it. So TN7 never moves out of this position until it finds a place where it wants to go. So it has various systems for looking for spaces that it wants to go. And when it finds it, it signals back, and only then does the element pop out and go to this new location. So this ability has been moved around a lot in different pathways to recognize good things. And that has the question, where would you go if you could control where you go? And that's what I'm going to be telling you about today. To do this, it uses these variants of these 5 proteins. So these are the 3 topics I'm going to go through. First, a little more basic information about this family and how modular it is. I'll tell you about one of these times where CRISPR-Cas systems were co-opted by TN7 elements so they can do their own self-programmed guide RNA-directed transposition. Some of the bells and whistles it has that maybe you wouldn't think of, but that introduces problems pretty quickly if you're taking these systems. And then finally, how we're adapting it to eukaryotes for many different types of organisms. And finally, I'll tell you about another 2 families or so of TN7 elements that have specifically evolved to recognize telomeres on linear bacterial genomes and the advantages it allows for them. So it was a really fun paper that we published last year. Cool. So the first thing that happens is the TNIQ domain protein that's common to these elements recognizes a place where it wants to go. So in the case I'm showing you it here, it has picked up this DNA binding domain. We call this large family. It's the largest family, the TNSD domain, that it recognizes this specific sequence of a good place to insert. TNIQ is then responsible for recruiting another TN7 encoded protein, TNSC, this AAA protein that forms a ring. And then this signal is used to recruit the transposase bound to the element. So only when this thing over here comes together does the synapse happen with the ends of the protein, allow it to be directed to the new site. Only when it detects all of that does it cause the joining events at the ends of the elements. And in most of these elements, the non-transferred strand is cut by a separate endonuclease, TNSA, to allow cut and paste transposition. So it removes itself completely from the site where it was, leaving a double strand break, and you have a direct insertion of this element into this new position. So after this evolved, what must have happened over evolution, and something you can see in genomes, is that it kept diverging to other kinds of elements. So where this was TNIQ, folks, to this TNSD binding domain, you can see evidence across evolution of pretty much every DNA binding domain that you learned about in bacteria has been picked up to allow it to recognize different sites, usually essential genes that it recognizes across many bacterial genomes. What we found just recently, but I'm not going to talk today, but it's on bioRxiv, we've recently found these elements have also started to bleed back and forth in archaea. Most exciting to us is also this subgroup, Asgard, that is the branch point of eukaryotes out of the archaea. So some of the most exciting examples and most complex archaea. as you might expect, instead of having these bacterial DNA binding proteins, they have ones from archaea, where the closest relatives aren't in bacteria, but are actually in eukaryotes like bivalves, insects, us. So it's been really exciting to see how these elements, once in this new background, have adjusted to the DNA binding domains in that. And you can see that in bioRxiv. We're currently arguing with reviewers right now. The next exciting one, and that I already alluded to, is how they've now picked up on 6 different occasions different types of CRISPR-Cas systems so they can use it for self-guided guide RNA-directed transposition. In this case, there's no DNA binding domain that TNIQ interacts with. It interacts directly with one of these CRISPR-Cas systems, and in all the examples, it does so in a different way. So it wasn't like it was easy to do. It was just such a good idea that evolution discovered it on 6 or more occasions, and one of these I'll tell you about today. So I'll go into a little bit of the nomenclature of these diverse CRISPR-Cas systems, but different subgroups over the years have been picked up in this way. And another thing I'm not going to talk about, but that's also in bioRxiv, from the first time, learning from these examples of how nature did it, we've been able to engineer it ourselves. So actually these family of TMPB proteins that are guide RNA-directed nucleases, and they're actually the antecedents of these Cas12 CRISPR systems, we've been able to engineer them so we have a much smaller system without host factor requirements, which we think will be useful for engineering things. That's on bioRxiv, but I'm not going to talk about it today. So I've been talking a lot about these TN7 family elements, and that is that it has these proteins that I've mentioned. So the transposase and this TNSA protein, which is part of the transposase, which is specific to this family. So what I'm showing you here is a family tree from our first paper of TNSA. And this is going to be a little bit of a roadmap as we go through these different elements and the different ones that I've found. So everything I'm showing here is kind of like TN7 in the sense that it has TNIQ interacting with a DNA binding domain to recognize one of different attachment sites. So either tRNA genes or competence genes, or different genes that it recognizes as it's picked up these new DNA binding domains. What I'm leading into now is the CRISPR-Cas ones. So another aspect of sort of the magic of how this pathway works and how it uses control in the most productive way as a transposon is really by balancing these 2 targeting pathways. So all of these elements seem to have a specific safe site that they recognize in the chromosome. We call it its attachment site, sort of like bacteriophage, but these are almost always downstream of an essential gene, so they can recognize a conserved target but not inactivate that target. This allows them to insert in this one spot when it gets in the genome without the risk of having to knock out an essential gene. So it's not going to change the color of maize in these bacterial hosts in that way because it always knows where it's going to go. And it's probably because of this level of control that allowed it to transpose at a much higher frequency than other elements, probably 1,000-fold or more higher, because it can get away with that because it's only going to do so into that one site. So it isn't constrained maybe by like some other elements. All of these TN7 family elements seem to have evolved other mechanisms where they can also recognize a mechanism for horizontal transfer. So in the case of the TN7 prototypic element, they have this TNSE protein that specifically recognizes replication associated with conjugation. That means they can recognize when the next bus ride comes into the cell, hop into that element, and then get transported to a new host. So by using these 2 pathways, they can recognize a safe site at a high frequency, but then also a mobilization mechanism to the next host. So they never have to be lucky about getting to the next host. They're really taking control of the whole pathway. And they're never not going to knock on an essential gene because by definition, this is new to this bacterium that it's recognizing as its ride to the new host. So what I'll tell you about some of these other systems is that every type of TN7 family element seems to have reinvented this in different ways. I don't know if you've heard this quote from Mark Twain, that history doesn't repeat itself, but it rhymes. And the same thing I think happens in evolution. Good ideas come again and again, but evolution discovers it in different ways to do the same thing. And I think that's one of the take-home message for this and exciting ways that this happens. So what I'm going to do now is get right into the CRISPR-Cas systems. I think everybody in general terms, and maybe their parents that aren't in science know something about CRISPR-Cas just because it's in the news a lot, but just to go into the details that maybe you didn't know a lot about. So maybe you're familiar with these immune systems and acquired immune systems of bacteria. So they have these three pathways. In the first phase, they have spacer acquisition, where they take a snippet of something like a virus or bacteriophage that did them wrong in the past but didn't kill them, and they keep it as like a wanted poster in their cells so they know if that guy ever comes back again. The way that they access this is they make a pre-CRISPR RNA that's processed into a ribonucleotide complex that they compare of everything else in the genome until they see that invader come back again. They then have a separate protein that can destroy it either by making double-strand breaks or loading a processive nuclease that can chew through that. So all of the various CRISPR-Cas pathways go through these same three steps. But the underlying systems themselves are very diverse. So there's 7 different types of these systems that fall into 2 major classes. One is the single component Cas9 that probably is the most famous of the bunch, but actually the way more common ones are these class 1 examples that have multiple components that do all of these things together. The whole pathway of this CRISPR-Cas system has been independently reinvented in these different systems. The only commonality is the spacer acquisition system that's encoded in these proteins Cas1 and Cas2. So it seems that once evolution discovered this, then it could kind of go crazy and keep reinventing the other system where it recognizes a protospacer adjacent motif, a PAM, and how this whole system that you're probably familiar with. So as I said, these systems were co-opted by Tn7-like elements on 6 different occasions. Some of these we discovered with Eugene Koonen's group, and some we discovered in our own lab, but we discovered all of these systems. And again, we have this recursive pathway of each way that they did it in every case. So one of them would probably be pretty obvious, is they had to get rid of the interference thing. So you can't cut the thing that you want to transpose into, right? So we have this recursive evolution where either they leave out or deleted the nuclease, which they no longer load onto there, or you find what looks like precision inactivation of the nuclease domain of that protein. Again, you can't cut the thing you're going to transpose into, so there's a reason why this must have kept having evolved. For reasons we don't understand, but we have ideas, they also lost the spacer acquisition system. So even though these systems are replenishing their spacers all the time when you look across genomes, they can't do it themselves. So they must be in a host that does it for them, and there's likely some benefit that we don't completely understand to making sure someone else does it and they don't do it themselves. So I'm going to talk about 2 of these, one we discovered last year, and then one that really is the best studied one, which we've been using for applications. So now I'm going to go a little deep into the nuts and bolts of how these interact and some of the questions that maybe are the second thing you would think of as you start to think about them. So the element here is from right to left, so everything between these moves. I've highlighted the CRISPR-Cas system and the transposon proteins, but what's indicated by double hashes is a lot of other stuff. So these guys are real pack rats about carrying other things around. So the most common thing they will carry are phage defense systems, things like that. But if it's one you find in a hospital, you can be damn sure it's going to be antibiotic resistance. And our most least favorite element in this category was this one in Acinetobacter that has 83 genes in it that 40 of them are encoding antibiotic resistance and biocide resistance, where one transposition event makes that bacterium resistant to every class of antibiotic that's ever been produced by humans. So they're very, very good at collecting very, very bad stuff. Good for their host, but bad for a lot of other situations. I'm not going to talk about those genes at all. So what they have in addition to these CRISPR-Cas systems is this guide RNAs that recognize things. These CRISPR arrays are often very, very small, where in normal CRISPR systems, maybe there's 100 or even more different guides that will be represented. For whatever reason, these guys are usually a handful of examples. But one of these, as I'll explain, recognizes their attachment site. So sort of that favorite site of theirs, where you will always find that protospacer that's recognized that adjacent to where you find the element. So this is easy to say, but it took us some figuring it out. What I'm also going to tell you about is this ability where they actually, in cases, have two types of guide RNAs. One that recognizes their attachment site, as I indicated here, but one that's always consistently replenished, which whatever mobile elements are in whatever neighborhood or environment they are. So to be able to move around, they have to be sampling their environment all the time by getting new spacers. But they also have to treat these two spacers differently. And this is the first example where we could see guide RNAs being categorized with these, and that's one of the stories that I'm going to tell you next. So a lot of this is bioinformatics, and we've really gotten to really use this a lot in our lab. In this one class of 1F3 elements, if you look across gamma proteobacteria where you'll probably find 10, 15,000 examples. This was an earlier capture that were deduplicated down to 90% identity or something like that. And what we noticed is that even within these 1F3 elements, there were two distinct families or subfamilies that seem to be behaving in different ways. One thing that was noticed early on is they did not have TNIQ fused to a DNA binding domain. So it wasn't that example that I showed you before that we see in TN7, but the whole process is encoded by guide RNAs. So they recognize the safe site in the chromosome, and they recognize the site in mobile DNA elements using two different guides. So they accomplish the two pathways, but all with guide RNAs. And this happens in almost all of these systems. What I've just indicated by red boxes are the two prototypes for these two groups that you'll see in the next slides. What are indicated here are the four major attachment sites that these guys are utilizing. So this is the safe site they use in the chromosome. So really the first thing that led us to this idea was this guy from I'm forgetting the name of it now, but these guys that were we could find one example that was in their safe site in the chromosome downstream of the FFF gene, and then a very closely related one that was in a mobile plasmid. And we could see that it had these two spacers, so it was a good test system for us to really prove what we modeled by what we were seeing in the genome databases. So what we did is we recoded all these things for E. coli, and actually I don't even need to recode them, but we expressed them in E. coli, and we had different targets. The target in the conjugal plasmid, the chromosomal attachment site target, or a target that didn't appear to have any spacer to it. And sure enough, we could see we got nice robust transposition into this target that was found in nature on conjugal plasmids, this one in the chromosome. But if we had a case where there was no target predicted from here, we saw basically no transposition. So this is that remarkable level of control I'm saying about. Even though we have 1 to 15% transposition frequency, you get almost no events unless there is a guide that recognizes that target. So 10 to the minus fifth, which actually might be 0, but the limit of our detection. So now I'm going to go through some of the nuts and bolts of these two families and how it's able to control these things. So again, the guides that are recognized, like the one in blue recognizes this one recognizes this. So they had guides that recognize these attachment sites, but we wanted to know how this process worked and also how it was regulated. Another important question that I'm going to thread in here before is because they have these guides from this active type of CRISPR-Cas system, they have to have a way of making sure the other system doesn't get their guides, because otherwise that will kill everything that they want to hop into. But this is a true love-hate situation because they also need to be in the same cell because that's the only way they get new spacers. So how that's balanced is made more clear in what I'm going to tell you in the next few slides. So each one of these guides I've indicated, we could find its protospacer match, and it's about 48 base pairs away from where we find the insertion. So we knew that was an important part of the process before we tried to reconstitute it. A couple things that I'll just mention later is, in addition, they showed more mismatches than you'd expect of native systems, and they also didn't seem to have a match to the PAM. So if you guys have ever set up a guide RNA-directed system like Cas9, you not only need to match your spacer or guide to your target, you also have this PAM, or proto-spacer adjacent motif, that is there. But they broke their own rule, but they were sitting on that site. So we were like, what's going on with that? And it is a sneaker ahead is that it turns out they're PAM ambiguous. And this also is one of the tools that allows them to have private guides that other systems can't utilize. So it addresses that question. So I'll tell you first about this A group here, which operates a little bit differently. So in these cases, we implicated this DNA binding regulatory protein, XRE family proteins, and we could find the motif that it recognizes, and it was sitting right in front of the guide RNA. So this was our first clue that this was somehow how it regulated access to the attachment site. That was held in a second array. So even though this was an array that was going over turnover for different kind of mobile DNAs in the element, this one was fixed and it had this regulator in front of it. This family of regulator was actually of a specific class that come from these, they're called controller proteins. You find them in restriction and modification systems, and all these XRA proteins have this unique behavior where they do zygotic induction. So if they find themselves in a new host, they express the gene very, very strongly. But after they're in some period of time, they tightly shut off expression. These were found in these restriction enzyme systems, but they were stolen on two different occasions, different family ones that were clear, where one gave rise to this larger B family and this to the A family. I don't know if you guys are familiar with this, but it addresses another problem. Restriction systems are often on mobile DNA elements, so they will recognize invaders of not having that methylation pattern and they'll cut it. But if you think of that restriction system comes into a new host, it's all foreign. There's no methylation on that genome. So these controller proteins allow the restriction function to stop and allows it to methylate strongly to cover the whole genome with methylation before it turns on the restriction motif. So it has to do this zygotic induction to allow restriction modifications to be mobile at a time, and most of them all are. So what it turned out is Tn7 stole this system, so it could also control this zygotic induction, something we wanted to prove in a simple system. So we put on an F plasmid the LAC-Z gene so we could monitor beta-galactosidase blue-white as an accurate way of looking gene activity. And we put it downstream of the site that's recognized by the XRE protein and the XRE protein itself. So we could show in one experiment that if this guy was just sitting in a cell with the XRE gene, the genes were off all the time, regardless of whether the chromosome also had the regulator. So if you look at these dash bars, the genes are just off. If we mated this system that had LAC-Z into a new host, now there was no repressor in there at the time, and we could see the telltale boost in expression of that gene at that time. So this would be the equivalent of the guide RNA coming on strong into a new host, so it can insert into the attachment site. However, if there was already a copy of the element in there that had the XRE gene, you would not see this boost of production of the guide RNA or LAC-Z in our experiment. So this showed to us that this zygotic induction that had been taken from these restriction systems was allowing to turn on that guide for the attachment site only when they came into a new host that probably helped protect it from other systems from being able to steal it. So this was a very satisfying explanation for these two array kind of strategies. But what we noticed in the second group is everything was contained in one array. So it couldn't really have that same explanation. So that's something we investigated further. Another thing is that the regulatory gene wasn't in front of a single guide RNA that recognized the attachment site. It was actually in front of the whole system. So it was a little different than the other one. So we needed an explanation. One of the things we noticed when we looked at these arrays is there was something kind of fishy about the repeats. And not just because they're fish pathogens, where the first two spacers actually had a true repeat. And actually for spacer acquisition, they have to copy the same repeat again. And that's what we saw in these first two repeats indicated with black triangles. When we looked at the spacer that was actually being used to recognize where they were inserting, it was hardly a match at all. Like if you look at this one, it's all T's. So we didn't even imagine that it was working at all. So in our initial work, we wondered, was this just a degraded one that wasn't functional anymore, or was there something else going on? And you can see the same thing here. So now we wanted to test what was the difference between the real repeat and this one atypical repeat that didn't even seem like it should be functional. So we set up a system again with these same kinds of guides where both of these guides would be made with their different repeats. So the repeats make these handles that are opposite the guide RNA part that recognizes this thing. So the machinery grabs these two 5' and 3' handles, and that's what it uses to look for where that guide RNA might match. So that's where the system grabs onto. And the question was, could it still be functional in that case? And the answer was, it wasn't functional, it was super functional. And not only were the atypical guides functional, but they functioned 10 times better than the actual repeats. So a bit of a surprise. So in the results here, we looked at reprogramming it for LAC-Z, just our favorite indicator. And we could find in the two examples, even though the guides had different frequencies, the atypical guide, so the one that was all like T's or U's, actually was working 10 times better. And that was true for the native target site, the FFS gene, also 10 times higher with this atypical guide. And it turns out if we made an exact match, it really didn't make much of a difference. So we believe there are two different ways that they're regulated. One is using the zygotic induction that is in the family A. And in the second case, these atypical guides that allow guides that are in the same RNA to be fundamentally and intrinsically different because the atypical one responds differently to the CRISPR-Cas proteins. What I'm not showing you, in the interest of time, we went on another paper with Liz Kellogg's group to structurally identify the mechanism for that, which is also very exciting, but it's published and I don't have time to talk about it. I'm probably going to run over anyway. Cool. So what I'll mention quickly, which is in the paper that I didn't get into, and sort of a repeat I said, for the one family, the A family, have this ability where they can only turn on the guide when they first get into the cell, and we think this is a form of protection from the native canonical system so they don't get degraded. There's also these atypical guides that work better in this system than the typical guides. What I didn't tell you too is that in addition to being really, really good with the transposon-based systems, they're no longer functional in the native system. So they're completely private from the other systems that might be able to kill them. There's also this mismatch function that is tolerated by the transposon systems, but is not functional with the other systems. So even if another system steals this guide, the mismatches are too much and it doesn't kill the thing they want to hop into. The other super clever thing is these mismatches are all focused at 3 base pair positions, and these exactly line up with the wobble position of amino acids. So it also future-proofs itself because it doesn't know what the third position is going to be in those open reading frames, and it doesn't care because that's the ambiguous position. So all of these things work together in these two kind of pathways to allow it to be functional in a variety of different hosts, but also protected from other kinds of systems that might otherwise be problematic. So that was the mechanism that this 1F3 system uses. It turns out another system I won't tell you that uses this Cas12 protein also uses atypical guides. But in these two examples, the way that they do two pathways is they have two complete systems. So they have a system with this TNIQ, TNSD protein that recognizes an attachment site, but then they have a second TIQ that interfaces with a CRISPR-Cas system. And this has happened 3 different families. One's not indicated here. So other ways that it's been able to address this same question. So I told you about some of these cohabitation mechanisms. I'm just going to do a couple slides on how we've been adapting this system for eukaryotes. So these things are rampant in bacteria, but we'd really like to use this as a tool to modify or have precision gene integration in humans for therapeutic things. We're working to do it in plants. We have a lot of collaborations. I'm going to tell you about some of our work in yeast. So this is just another diagram of the CRISPR-Cas system, TNIQ and C, and how it's recruited to a site. Again, these are all in bacteria, so there is no nuclear localization sequence. So we needed to deal with that as a thing. We also, if you're going to express it in eukaryotes, you want to have as few components as possible. So we found that we were able to fuse TNS-A and B, and they were functional, maybe even a little better than having the protein separate. We noticed that having the NLS on the N-terminus or C-terminus here seemed to be problematic. So we just stuck it in the middle, and that also seemed to work, and it allowed us to be able targeting in that case. We then added nuclear localization sequences here, and that seemed to work just fine. We got the same amount of transposition with the NLSs as in the no NLS examples. The only protein that was problematic was TNSC, and we noticed this slight drop when we put an NLS on that. So for that, we took an alternate strategy, and we tried exposed domains within the protein to be able to tag it. And we found one that actually allowed a little bit of a stimulation and also a little bit of TNS-ABC independent transposition that we also used to look at more systems. So this was the NLS tagging here that allowed the system to work that we then wanted to port out to other systems. Oh, yeah. So one of the other things we've done is an old sort of trick that was for the initial CRISPR-Cas systems is they really looked across different evolutionary examples of them to find the best ones that would work in eukaryotes. So we looked broadly and picked up 40 examples across the whole family of these CRISPR-Cas elements to make sure we were sampling for ones that might do best in eukaryotes. So we tested all these in this minimal system where we had the internal NLS that seemed to allow it to function at a low level on its own. And we got a distribution. So although this was the frequency of our original candidate, we found many that were much higher in that. And then some of these we went on to test to see if they still responded to their guide RNA systems. And sure enough, they did. And these examples, as we had hoped, were much higher than the native system, so almost 100-fold higher. So we're using this as our pool of examples to test more broadly in eukaryotes. And I'm just going to show you one example here where we've adapted it to yeast. So as a way of monitoring transposition, we actually initially screened by not where it went, but that it left. So these are cut and paste transposons. So when they leave, they'll leave a double strand break in their target. We set it up such that repair could fix it, and we would get an active uragene. So if the element is in the uragene, it can't grow without uracil, but we could select without uracil to find the transposition events. And we got a nice low level of transposition with many different systems. And this also allowed us, because this doesn't have the targeting component, to look to see what was accessible. So we had some fear that the chromosome may not be accessible to this prokaryotic system with chromatin and everything like that. But what we could see is all 16 chromosomes could act as a target, and we found the insertions in both orientations indicated as red and blue. Not shown here, but the vectors that were used in the experiment and even the mitochondrial DNA, we picked up insertions that map to all of those locations. So it seems like it has access for our future work as we try and set it up for targeting. So 2 years later, we've now been able to reestablish targeting, but it was not very simple to put the CRISPR-Cas component into yeast as well. It turns out it's a little bit toxic. Okay, I'm not going to present here, but another thing we've been doing is we've been mutagenizing the components to kind of evolve it in the laboratory so we could have higher activity systems. And we're easily getting 5, 10 times more transposition, but I won't go into that. But that's sort of an exciting way that we're trying to really get the best systems by having examples from the diverse elements and then also being able to get these gain of activity mutations. So what I want to talk about now is this family of elements that are a bit different, but also have a preferred target where they recognize telomeres in linear genomes. The first example we have came from a weird branch of these Cas12 examples of guide RNA targeted transposition, where instead of having the CRISPR-Cas system, they were missing it, but instead had a protelomerase, which was really weird when we started. So what is a protelomerase? Most people don't even know that there's linear genomes in bacteria, but there are quite a few, and there's two major mechanisms that allow to do that. And one mechanism, they put a hairpin at the end of the DNA. So if you have a linear genome, there's two things you got to worry about. One is the end replication problem, because you can't set up your lagging strand in that case. Having a hairpin allows you to just replicate around, so that solves that problem. The other issue with the telomere is you don't want it to look like a double strand break, because you're always trying to fix a broken end that isn't broken. So by having a hairpin, it actually accomplishes that second thing. These things aren't particularly rare. Lyme disease has this, the Borrelia. So a true plant pathogen also has this on one of its chromosomes, Agrobacterium, and there's a number of phages that have the same kind of a system. How we discovered these things, although there were many, many examples where we had this system next to Cas12, we found relatively close homologs that didn't have Cas12, but instead had a protelomerase. When we look at these, they seem to be at the end of contigs, so that could mean anything, right? But in many cases, we could show that we had the remnants of the very end, this hairpin that when you sequenced it, artifactually it'd have a inverted repeat looking at the end. We also found that you only found one end. Even though two ends are needed for transposition, we were only finding one, and they had this unknown small ORF. Yeah, these guys were, the same family. So these are all these Cas12 elements indicating the protelomerase elements that I call PAST. They seem to be no different with some of the proteins like TNSB and C, but in the case of TNIQ, they seem functionally diverge. And this underlied this idea that maybe the Q adapted for something else that wasn't Cas12, but instead the protelomerase. So we wanted to test this model that through the normal pathway of these telomeres, when you have replication across the hairpin end, you get this, double chromosome at the end, and the protelomerase brings it back to a single so that it could be the normal single chromosome. So our model for why they only had one end was that transiently this double strand end would give it a full transposon. So the two ends needed for transposon, and it could move that telomere and take over another telomere in the cell by doing their transposition process. So we need to show that this telomere, this protelomerase actually recognized this telomere. And we could show that biochemically. I guess in the interest of time, I'm not going to go through, but we could show that it had a hairpin and that it specifically recognized this end. So something I'll come back to in just a minute is that in addition to recognizing a telomere, it also steals the telomere because it adds a new telomere that's only recognized by its own telomerase. So the host cannot get rid of the transposon without killing itself because it would have no means of maintaining its telomere. So this allows them to be addicted in their new host. It's a very clever way that they can capture that end and can't be lost. Again, in the interest of time, we could show that all of these components were needed and that they actually seem to have this ability to target linear ends. So amazingly, in E. coli, even though everyone probably knows it has a circular genome, you're able to clone in one of these phage linear systems, and you can make the E. coli genome linear. And what we were able to show when we analyzed transposition in this system, we could find that it required all of those proteins, but that when we compared a circular native genome to this non-native linear genome, transposition was repeatedly a bit higher. And we were able to show that even though in both cases there were a lot of random insertions, you could also see insertions accumulating around this linear end within a few 10 kb of that linear end. We have ideas but don't understand how this happens, but it seems to be able to recognize linear ends directly. In a way, it has to do this because whenever it transposes, it brings a telomere with it, and whatever DNA it recognizes, it makes it into a new telomere end. So you might imagine if it inserted in the middle of a genome, it would be a very bad situation. So it kind of has to recognize telomeres, and this gives us a hint into how that process works that we're working on. So yeah, this is the whole pathway that I had told you about. And most of these steps is very much like these 5K CRISPR-Cas transposon elements, but it recognizes telomeres instead. As I said, there's a lot we still don't know about this, but it does act as an addiction system in that when it moves, it makes wherever it integrates into a new telomere, stealing the existing telomere from that host. So it has a way where it's able to hold on to those. So I'll tell this story really quick because I think we're probably getting close here. But the second thing we got interested in is like, hey, we found these guys were able to target telomeres in this hairpin example. There's a much bigger family of elements that are native to streptomyces that also have linear genomes. Can we find these elements there? So streptomyces genomes tend to be very large with the core genomes internal to them. But they do have these more flexible, rapid turnover ends that often accumulate a lot of mobile elements near the ends. These ends are also on a lot of the linear plasmids that these guys have. So the way these systems work is they don't form a hairpin. It's they recognize a sequence at the end with this TPG protein, which then binds to there. It has a second protein TAP that's a polymerase that uses a threonine to make the other strand. So by doing this, they solve the end replication problem because they filled up this gap that couldn't be filled because of lagging strand replication. They solve the second problem in that it doesn't look like a double strand break because they're covalently bound to that DNA. So it was just another way of solving the same problem that is extremely common in most streptomyces. Oh yeah, so it turns out these are also mechanisms that allow these things to be transferred via conjugation. So that's why mobile genes are often at these ends that are exchanged between hosts. So the way we look for these is predicting that the families we were interested in should be near the end, and they should also have some kind of an orientation bias. So we looked at all the transposases across streptomyces, and we just look where they mapped relative to the centromere and telomere with the idea that they should be at the telomere as our thing. So probably with blurring your eyes, you could say all of them are a little bit biased, all the different families to the telomere, but there was a few that were just smoking guns that were almost always in one orientation and very close to those ends. So these are the ones we became interested in. So these ones have the full A, B, C, and Q, and these are minimal Tn7-like elements or mu-like elements that have B and C that we got interested in. So first is looking at this family here where we have a tree of the TNIQ gene on all these examples. So the way to see if these guys were really in this family of elements, in addition to having the TNIQ tree, we mapped on these proteins that were important for telomere maintenance with the idea that were they also mobilizing the telomere maintenance, and did they also steal telomeres from the native host? And the answer is they do. So these bars here indicate that they have in these two families these same kinds of end maintenance proteins. In addition to that, we could find not that only they have it, in that graph they're within 60 kilobases, but they're also like nestled in between the transposition genes, an indicator that they were responsible for actually being in this protein all the time. So where the transposition genes in pink, the telomere maintenance proteins were nestled in there, and this is one of these indicators that they are involved. I'm going to go over this point too. It turns out that you cannot be a cut and paste transposition if you insert into a telomere because you cut it off, you guillotine it. But these are cut and paste transposons, so it was a bit of a question to us how that actually functioned because the TNSA activity would cause this break in the non-transferred strand. What we were able to show is specifically those telomere targeting TN7-like elements had inactivated their active sites for TNSA. So there should be an E, D, or RD that was native to these elements, and that was found in all the other TN7-like elements, that those that specifically recognize telomeres, as it turns out, it was inactivated. So it doesn't seem that they do this, and this solves them from cutting off telomeres. So the very last point I'm going to make is this other family that only had TNSB and C. And it turned out these elements, we could see that they had these two-ended relatives that were sort of like a basal branch that had two ends and then hopped like a regular transposition, a regular DNA transposon. But all these other ones seem to lack those features that we looked at here. So there's more questions than answers in here, but things we're interested in. Some have this MRE11 homolog. It's a end recognition and repair proteins from eukaryotes and bacteria. And somehow we think that's allowing it to recognize telomeres. We have this other group that have dedicated telomere maintenance proteins close in. And then finally, this other CRISPR-Cas system. This was this new one we found that we think is being used to cut telomeres to use them as targets. So unlike the native canonical E systems, they mapped separately. So if you looked at ones associated with these transposition proteins versus the others, they all map differently on their same branch, whether it was the Cas3 or Cas7. If we looked at the spacers they maintained, even though native canonical defense 1E systems not associated with transposons could have spacers all over the genome, the ones that recognize telomeres only had spacers that recognize the telomeres themselves. So we think somehow they're breaking these ends to use these as integration sites. If you look very, very carefully, we can find some examples where the protospacer match can be found in the telomere region at some distance away from the point of insertions. So I won't go into this in detail, but we can also prove that these guys are actually transposons by making a make-believe end that instead of having one, we artificially in E. coli put two ends on it. And we can knock out regulation by disrupting the Walker motif and show that we can get transposition with all the hallmarks of this kind of transposition, like the target site duplication that you expect as part of this process. So we do think this is a new bona fide family of Cas elements, so CRISPR-associated transposons, that targets the telomere somehow by making a break and then some unknown mechanism recruits the transposition, but it must be different than the other kinds of elements. Yeah, so that was the last data side. So we found these many different families of CRISPR-Cas elements, and sort of a backdoor from looking at these, we also found these telomere elements where they're able to accomplish the same thing. They can recognize a safe site they're going to find, so in this case, the telomeres, and they also have an insurance policy in that they replace the telomeres in all these systems because they can't be lost. They now own those telomeres, and they're kind of locked in there. It also gives them an access mechanism for getting between bacteria, because I mentioned very quickly, these TTRA systems are the conjugation regions at the ends. They get moved between bacteria. So they also have their ride to new hosts as they go around. So these are some of the people that we worked on. Cornell has also filed a number of patents on our behalf, and we worked with some companies. We'll start one this summer. These are the individuals. Much of the work was by a guy who goes by the name of Popo and Mike Pathology for the first story, but then the work in modifying for new eukaryotes has been with a lot of people. Here are some of the structural biology collaborators that we've had in this work. So happy to answer any questions you all might have.
Any questions from grad students or postdocs first? All right, Brian, go for it.
Yeah, I think it's really amazing that your TNS-E and D pathway, you have both of these mechanisms, that the CRISPR system developed the same strategy to have the safe site and the mobile DNA. So how do you think that they made that jump?
I think it was just like a good idea. Like you want to be able to find a safe site that you can hunker down in a host, but what good is that? It's like the thing that was the advantage becomes the liability, because then you have no random transposition that would hitch your ride to the next host. So I think you almost couldn't have one without the other.
Not all transposons do both mechanisms, though.
Right, but yeah, I mean, we think there are elements that are random, but most transposons will just move all the time. They don't have this mechanisms. These family elements only move when they find a place they want to go. But that is a huge advantage because it allows these kinds of things. But I think you have to have both. If you recognize something specific, you need a way to recognize a ride to other bacteria.
I have a question. So in filamentous fungi, there are these like giant transposable elements. I was wondering, like, if you're like trying to move stuff into eukaryotic systems, if you've like done anything with like trying to utilize some of these like native systems and combining them with.
So almost none of those systems, actually none of those systems have this property, you know, because a lot of people have taken eukaryotic transposons and they just stick a CRISPR-Cas system on it. And you can get a small number of RNA-directed insertions, but you still have all the random insertions. So in their title, it'll be like, guide RNA-directed transposition, and they leave out, and all the random ones too. You know, like, it's a loser strategy if you want to avoid those other strategies. So I have seen nothing yet of like that. There's this, and this is a little bit of a tangent. I don't know if this is Keith Slotkin guy. I don't know people who do like gene editing in plants, where he worked up a CRISPR-Cas system with, it was the ping pong elements. And there you could get guide RNA transposition, but the way that it functioned is that they didn't work together. The guide RNA system was making breaks, but the element just excised and it repaired the break. So it's not like a true guide RNA system, but that's the only example I know where something guide RNA has been engineered in this way.
So I've been reading a lot about like retrotransposons, some of the filamentous fungi that I work with, and a lot of people are sort of starting to develop what the genomic architecture and evolutionary history of the transposons look like. Have you done any of that in your transposon?
So the evolutionary past of these elements.
Point did they sort of start to engage in the genome?
It's a really hard question. You know, this is the first time where we can show that they moved between archaea and bacteria, and it was at least 10 different examples, but we don't even know what direction they went. You know, probably where they're more diverse, they probably started in bacteria where they're very broad, and their penetrance is very variable too. So overall, about 10% of all bacteria, if you look in mass, have it in it. But then there's some groups like Streptomyces where 40% of the genome have at least one example. 17% have one of the families of telomere, 17 have the other. So Streptomyces is a wash with these guys. The telomere targeting, and also in cyanobacteria, for some reason, they're very enriched. 40% have these family elements, and 5% are the telomere targeting ones in cyanobacteria. But like proteobacteria, for whatever reason, it's 20%. So they're around, but they're not 100%. And they're always dying too. You'll find many remnants of these elements in genomes as well.
Any other questions?
Cool. Thank you very much. Yep.
This has been a production of Cornell University, on the web at cornell.edu.